All right, so we start now. Thanks for coming in this fantastic day. We really excited just to go and do a little bit of cycling. Anyway, thanks for coming. This is the last event of the Art Salon series. My name is Roberta. And together with Stephen, who could not be here, but imagine that he's here next to me. We are co-curating the Art Salon, which is a series of events, and you'll hear more of that, which tries to connect artists and scientists. Throughout the year, we had all sorts of events. We had stuff about data, stuff about icicles, about geometry, maps and territories. We had neutrinos, the big protagonists. What else? We had a lot of really science and arts related events. So this is going to be the last one, and we're going to talk about spaces of science, what counts as a space of science. And we are very fortunate today to have two artist scientists who have been kind of disrupting the idea of scientific space. One of the questions that we are asking these days is how can we define scientific space? Is it necessary to be in a science lab in order to do science? Or can we do art in a laboratory? Can we turn an art studio into a laboratory? Or can we create a laboratory in our garage? So we're going to answer all these questions, hopefully, and engage in a conversation with Nicole Clouston, who is a bio artist, who is also doing her PhD at York University. And with Jasmine Alkin, who is the president of the DIY Biology lab here in Toronto, which is 2 years old. 2 years old, yeah. So it's kind of new, but not very new. And both of them will talk about their own work, and eventually they will come up with questions and with answers and with some discussions. Just before we start, so this event is recorded, and everything will be archived afterward. Some people probably are listening from wherever in North America. And I have to say that this event was, we're very grateful for having all of this technical support and this fantastic space. The Fields Institute is the host, and we have been sponsored by the Ontario Arts Council for throughout the year, and we hope next year too. Anyway, let's start. So, Nicole.
Hi, everybody. I'd like to start just by thanking Roberta for organizing and inviting me to speak today. So I am going to be discussing my practice as an artist and how I came to work with microbes, so delving into this more scientific avenue of working as an artist. And I'm going to talk about sort of why I began down this path, which sometimes seems kind of unusual for an artist to go down in their studio work. I always have had an affinity for science, and largely as I was growing up because of its perceived capacity to provide answers to these huge questions that we all have. And this interest has continued throughout my life. And I'm often really inspired by both the wonder and the terror that I feel when investigating scientific discovery. So one such point of inspiration, of awe and terror for me, has been normal flora, so the ubiquitous bacteria, molds, and yeasts in our daily environment. And I began actually first engaging microbes through representation. I was really drawn to images I found of gram-stained bacteria. I found them incredibly beautiful, and at the same time was really questioning my interest in them because they were of these horrific diseases like MRSA or other antibiotic-resistant diseases that were cropping up that were obviously causing massive damage to the human body, and yet in images like this were quite seductive. So I started engaging with those through a drawing practice. And not necessarily replicating a specific bacteria colony or virus, but taking inspiration from the forms that I was seeing in these gram-stained slides. Yes, thank you. So after I'd been doing a few of these drawings, I started realizing that I wasn't finding it particularly engaging. I wasn't satisfied with the fact that I was merely representing an image of the microbes. I wanted to actually work with them directly. And this kind of came about as I started discovering this field of bioart, which I had not been familiar with before, and particularly the work of UK-based bioartist Anna Dimitriou. So through her journey of becoming an artist who became really involved in microbiology and has gained access to really high-level labs, I sort of realized that there is a possibility of an avenue for an artist to directly work with these types of specimens. And after I decided that I really wanted to work in this manner, the biggest challenge then became how. How am I going to go about gaining enough knowledge as an artist with very little scientific or technical knowledge in terms of this field? And so my biggest resource at first became the blogs of hobbyists. So people just working with scientific experiments at home, very simply engaging these kinds of microbes. And also scientific papers, as well as the wonderful biology faculty at the University of Victoria where I was studying at the time. And so through just looking at these various blogs, I found the Winogradsky column. So that's the image you see here. And the Winogradsky column was invented as a way to grow microbes in concentrations far greater than you can just find them in mud. So this technique was clearly something that I could do. I felt comfortable approaching this. And so this was my starting point of working with microbes, and has sparked actually an ongoing project of mine, which is provisionally titled Mud. So the piece is composed of a series of clear acrylic prisms, which are filled with mud, as well as nutrients which encourage microbial growth. So that would be cellulose, which I put in the form of newspaper, sulfur in the form of egg yolk, and calcium carbonate in the form of the egg shell. So very simple materials that come together, and then through exposure to light, allows the microbes already present in the mud to really flourish and create vibrantly colored bands throughout the sculpture. And the artwork itself forms a self-sustaining ecosystem, which is something I'm really interested in as well. I'm able to put the mud into this prism, feed it once, and then allow it to grow on its own. And it continues to shift and grow throughout its lifespan. So as I'm showing you images, you'll see sort of slow shifts in the ways the microbes have grown in the containers. And although the microbes in this sculpture are not directly from our bodies, they do call attention for me to the trillions and trillions of microbial life forms that are sharing the planet with us, that are in our bodies, on our bodies, and in our environment as well. And I also found that this work revealed to me, and hopefully to viewers as well, the beauty of these microbes, which we often have a fairly negative perception of or relationship with. So attempting to foster a different perception of bacteria and microbial life. And this is a more recent iteration which has not begun to grow yet, but sort of revealing the evolution of where this work is going as an ongoing project. So at the same time that I was beginning to work on this project, so when I was first working with microbes, I also started working with the microbes directly from my own body. Oops. Sorry, that should be playing a video. But anyways, bacteria in one's saliva. And so, again, I began working with my own saliva as a source of microbial life and culturing it in petri dishes as well as onto found mannequins. And this was my very first attempt at really exteriorizing that interior microbial life that I had become so fascinated with. And I really thought of it as a direct way to kind of confront the microbes that I knew were within me, but I couldn't see or engage with in any tangible, material way. And so I did culture it onto a human figure, again, thinking of my own figure. This is an installation shot. And all of this work has a lifespan. So for the mud, it's quite a long one. For the agar and bacteria, it does dry out and desiccate over a period of a month or so, and cracks and dries out, and the bacteria will slowly die. So there is a process of a life cycle also within the work that I'm quite interested in. Also, while I was working on these works, I was thinking about isolating my own microbes, right? And exteriorizing the life within me. But what I quickly came to realize was in a studio environment, I wasn't able to get a sterile enough situation where I could know that I was isolating those microbes, where I could literally just be culturing from my saliva. I was culturing from the fabric as well. There were bacteria that were growing that were just already present there. Obviously, also atmospheric, molds, yeasts. So I started realizing the lack of control I really had over what was going on. And instead of fighting against this and trying to make my studio into a pristine lab environment, I became actually really excited about it and engaged with the fact that I was encompassing all of this life. And so, again, a short video, but skip over it. In my most recent project, which was exhibited in the Art Gallery of York University vitrines this past January, I really embraced this aspect of the work. And I titled the piece Normal Flora. So again, thinking about just all of the bacteria, yeasts, molds that are in our environment naturally. So I thought about this work as making visible the invisible presence of microbial life within the vitrines themselves. So there were 3 separate vitrines, each with a sheet of gauze dipped in nutrient agar. So exposing the microbial life that was in the vitrines, as well as already on the gauze, and from my own body, just placing the work into the vitrine. So that all of that exposure began to grow on the sculpture's surface. So again, it certainly has a life cycle to it, starting out as a very fresh, kind of just moist-looking piece of fabric, basically, and then slowly the microbes will start to appear, and then also begin to dry out and desiccate. So I thought that by revealing the microbes already there in the space and part of our environment, I hoped to be able to provide viewers with that same experience of confronting the microbial presence that they were constantly passing. So the work was in a concourse, right, where people at York University, where students would be passing by every day and not necessarily considering what was there, right, this invisible presence that's constantly around us. And so I consider this a point of reflection upon that life. And a way to, again, really physically and embodily confront those microbes and consider them. So while working on this piece and previous iterations of it, I think I've already expressed this a little bit, but began to get a deep and visceral understanding of the proliferation of microbes in our environment as well as in our bodies. The fact that, again, we share the planet with trillions and trillions of these life forms, invisible to the naked eye, yet completely vital to our own bodily health as well as the health of the environment. And these microbes form vast and complex communities that are incredibly fascinating, that are around, within, and on our bodies. And just coming to acknowledge the fact that we actually have 10 times more microbial cells than human ones, and that these microbes pass in and out of our bodies, permeating what we like to consider the barrier between us, what makes up us, and everything else. And so when confronting this knowledge, I once again was awestruck, as well as completely terrified and grossed out and freaked out by the fact that what I thought made up me was not what I had expected or maybe hoped as I was coming to understand this knowledge. And so I became fascinated with this feeling and came to understand it as an experience of the microbial sublime, which is a focus of my current research. And my conception of the microbial sublime has been largely informed by the bacterial sublime, which is a term coined by Anna Demetriou. In her 2012 paper, Confronting the Bacterial Sublime, she states, quote, "There's a sense that the world is heading towards a new pandemic, that an unknown disease will emerge, or that an existing pathogen will evolve strategies to resist our limited antibiotic cures and strike us down. At the heart of this, a sense of the bacterial sublime, combining terror and awe as we reflect on the impact of these minute life forms, whose complex behaviors we are only now beginning to understand," end quote. So I was really fascinated with this concept, but I didn't feel like this was the whole picture to me. So while contemplating, I began to think of it less as the bacterial sublime and more as the microbial sublime. So in doing that, I wanted to shift attention away from sort of this fear of microbes, fear of infection, or antibiotic-resistant disease, and towards this symbiotic relationship that we have with bacteria and microbes. And also to include bacteria, but expand our understanding to viruses, molds, and yeasts as well. So all life forms that you are too small to see without a microscope, I think, have this capacity to evoke the sublime. And as I've been thinking about this, I've sort of thought of different ways that the microbial sublime can be presented to us, or an experience can be triggered. So one way is through very simply the visual presence of microbes, such as mold growing on the surface of coffee, or physical symptoms, like when we're infected with the rhinovirus or the common cold. And another way, which is sort of the way I first experienced the microbial sublime, is through new knowledge. Learning things such as the fact that on the tip of one of our fingers, there are more microbes than there are humans in the world. Things like this kind of boggle the mind and result in a disruption of ordinary perception, triggering, again, a really deep and visceral understanding of the microbial presence within and on your body. What I personally found and continue to find most interesting and significant about the microbial sublime is not just the fact that microbes have this capacity to provoke terror and awe, but actually what can result from that experience, and the potential to shift understanding after experiencing the microbial sublime. So psychologists Dr. Keltner and Jonathan Haidt actually argue that awe, or the sublime, is a really powerful trigger for change in human behavior. because of its ability to cause what they term accommodation, which is basically the process of adjusting your way of thinking or mental structures in order to assimilate or accommodate a new experience. So adjusting to things never before known or fully understood. And awe challenges these mental structures when they fail to make sense of what they're experiencing. So coming to understand these facts about our body and microbes, that we are permeable and porous rather than, a closed off system, can be a knowledge that, if you really deeply come to understand it, can be quite hard to grapple with. That our skin is not where we end, and the definition of us is much more complicated than it first maybe appeared. And they argue, again, that experiences of the sublime such as this can transform people and reorient their lives, goals, and values. Going on to even assert that awe-inducing events may be one of the fastest and most powerful methods of personal change and growth. So I've been really inspired by these potential implications, and I am continuing to work through the work that I showed you previously, through mud and normal flora, to attempt to engage the microbial sublime and working on making microbial life visible in a way that encourages these embodied experiences and potentially could evoke the microbial sublime for the viewer as well. For me, an experience of the microbial sublime really confronts us with the knowledge that our bodies are not closed-off systems, that we are porous parts of our environment. And by accommodating this awareness, we recognize our interconnectivity with the world, the fact that we are literally enmeshed in our environment, and that any separation we perceive between us and our ecosystem is completely arbitrary. It's possible that by acknowledging this really deep bodily connection with microbes, and by extension our environment, that we may begin to foster a stronger, more sustainable, and empathetic relationship with our ecosystem as a whole. Thank you. Switch over.
In the meantime, if you have any question, otherwise we're gonna just discuss later with the two of them.
Can I ask a question for this?
Yeah.
Hey, it was a very good talk. In terms of the, you were extracting something which is alive and then you are kind of isolating it.
Mm-hmm.
To go through a decay cycle, and probably that's not the representation which is, part of our body. Like, how do you...
I think, are you talking about the column that I showed? So that is...
The mannequin structure.
Mm-hmm. Yeah, it's definitely, I completely acknowledge that it's not how the microbes would live within our body, but it's a strategy, a very simple strategy that I can employ to exteriorize them. So to take them, yes, out of the way they would behave within me, and to try to see them, just to confront that presence. And the column actually isn't actually the microbes from our bodies, it's the microbes that are in soil. Yeah, but similarly, no, they wouldn't grow exactly in that manner. They would grow in far less high concentrations, which is why we don't usually see that type of vibrancy in the mud. Yeah. Thanks.
Can I ask you one more question? While I'm talking about mud,
Sure. Yeah.
is it different mud in each cylinder, or is it all the same mud?
So the way we're working with it so far.
It's okay. The way I've been working with it so far is that each series will be from the same water source. So I'm coming up on working actually on a project that will sample various parts of Lake Ontario. And so each spectrum will be from a different section of the lake to kind of try to see the variance of the bank versus deeper into the lake, etc. But also, it's interesting to see that there is such variance between even the exact same bucket of mud in different columns based on minute differences, probably in microbial presence. Yeah. Yeah, this is the thing. I'll move this. Yeah, you just plug it in.
I can do this off. Is it off? Just gonna put it.
Yeah, just stop. Yeah.
Okay, so I'm not sure how many of you here know me. I'm going to guess probably less than half. But as Roberta said, and thank you so much for asking me to be here, my name is Jasmine Alkin. I'm a graduate from McMaster University from Life Sciences, and my interest lies in genetics and molecular biology. So what do I actually do? So why am I even here? Well, I'm actually the president of an organization called Do It Yourself Biology, a place that kind of wants to open up science for everyone. I'll actually tell you a little more later about that, but first I'm actually going to start with what's science, what is art, and what are the similarities and differences in them. And of course, how we can use science to create beautiful art. So first we start with the powerhouse of the cell of the mitochondria. It's a stupid pun, but I kind of adore it. So let's start with science. First of all, science is, I'm going to be really frank with you guys, real intimidating. The language is foreign, the cutlery is really weird, and the pets are kind of gross. It's foreign, it's hard to understand, and it's more like a black box for the average person. So let's start with the strict definition of science. Science is the knowledge about or study of the natural world based on facts learned through the experiments and observations. That doesn't really tell you much though. So what do scientists actually do? We like to look at things. So we look at things, and then we say, I don't understand what this thing is. So we say, I have an idea. I'm going to try to explain it. So we hypothesize. Once we have an hypothesis, we go, okay, I need to prove this is right, and we design a set of experiments that will generate data for us. And then we actually start doing the experiments over and over and over and over until you decide to change your career. And once you're done and hopefully have enough data to actually prove your hypothesis is correct, which, usually doesn't happen, you can publish it. Why is this important? We need this to have inventions. We need it for medicine. We need it for physiology. We need it for technology. Without this level of proof, without having the standard of proving things and making sure they're right, we don't have what we have today. We don't have technology. We don't have computers or delicious food sometimes. So it's extremely important for everyday life. How about art? So this is a strict definition of art. It is something that is created with imagination and skill and that is beautiful and or expresses important ideas and feelings. I think that definition is kind of a letdown and doesn't really tell you what art really is about. So in the big grand scheme of things, what is art really? Well, art is an expression or the self-expression of concept, ideas, the self, the world around you, and how you interpret it. And why is art important? I'm sure for the artists in the audience, you always have people like, art is not even an important thing, no one cares about it. Well, of course, they're wrong. Art is important for, in my opinion, the strongest thing is that they communicate. And the way they communicate is a kind of communication that goes past the boundaries of cultures, languages, backgrounds, economic status, and health. And being able to communicate that way is essential for human society in order to relate different ideas to different people. So how do you combine two subjects that are so different? I mean, I just told you science was incredibly boring and incredibly repetitive and, not much happens. And I just told you art is very, diverse and lots of things happen, but not very, repetitively. So really, what does it have in common? And who's actually done art and science? So let's start with an old guy, Leonardo da Vinci. Most of you know him because he did the Mona Lisa and the Last Supper. He's an artist. He is actually known as a polymath, someone who is talented and knowledgeable in a variety of studies, including mathematics, physics, optics, and zoology. And he's well known in the science community for his contribution to anatomy. And around 1508, he dissected his first body, a 100-year-old cadaver, of a man who died extremely happy, apparently. And within 8 years later, he dissected 30 more cadavers. And as you can see, he had created extremely detailed drawings of what he made. And if we move on to the present day, of course, you have Nicole. And since she studied chalk, I won't go too much into detail about it, but this is the piece I liked most when I went to her website of one of her mud demonstrations. For me, I honestly don't know much about mud, but the colors and the contrast, I thought that was absolutely gorgeous. And the next modern day artist scientist would be Greg Dunn. Greg Dunn is a researcher, was a researcher from the University of Pennsylvania in neuroscience. But he has since moved on after finishing his PhD because, no one chooses science, and moved on to create beautiful art. So this is actually a cross-section of the spinal column, and it visualizes the nerves and its interactions with each other. And he actually does a lot of metal etchings and gold leafing, and this is how he created this piece. So let's go on to what they actually have in common. I honestly think they're more similar than different, because both artists and science aspire to understand the world. They want to explain concepts and what they're feeling. They want to invent new ideas, and they also want to inspire others to follow behind them. And I'm going to attack each subject very, very specifically with pretty pictures. So first, let's start with understanding. I'm not sure how many of you know, but this is a protein. And this is more or less what we think they look like. In this picture, you can see is that I have a cross-section cut out, which shows you sort of the ribbon pattern of the protein, which is kind of like the bones of it. And on these slides, you actually have the filled-in pattern of the protein as what it will look like in real life. It's actually from something called The Inner Life of the Cell, which is a viral video series that came around in 2009 that was very, very popular among, majorly popular among scientists, as well as popular among everyone, because it showed beautifully how everything worked together. The next one is actually a piece I found for an individual from the Ukraine who goes by the name Sally's Lips. He created this using something called fractals. Fractals is a recursive repeating pattern of the same thing over and over and over again over a large, large period of time. So a good example of fractal would be cauliflower, trees, basically anything that pretty much naturally grows. And he uses this concept to make a beautiful piece of art, which looks kind of like a flower with seeds in it, but that's kind of just my opinion. They also both use science to explain. So here we have an actual mitochondria, probably something that was decades of research to get to this point. And it kind of shows you the structure of mitochondria and what it does and the functionality and how it works to create energy in your cell. The next one is I'm assuming most of you know who Banksy is. He is a British street artist who's known for his political and sociological commentary. And here he drawn, of course, a bear eating what appears to be a dead fish or a skeleton fish, and as a commentary on the environment and how we're treating it. Inventing. So in science, this specific example is really popular. So at some point in time, when we discovered diabetes and discovered the treatment for diabetes is insulin, we started giving diabetics insulin. But in order to give them insulin, we had to get it from somewhere. At first, our source were pigs, and we were giving them pig insulin or cow insulin instead. The problem with that is that eventually they developed an immune response to it. Once they develop an immune response, you can't give them that insulin anymore. So instead, what the solution was to make recombinant bacteria. That means transforming bacteria to express the human insulin gene and extract it from that. This led us to be able to treat diabetes effectively over long periods of time without adverse physiological reactions. For the artist, we go back to Leonardo da Vinci, and we go to his helical air screws. So this probably doesn't look very familiar to most people. It looks kind of like a toy, actually. But this is pretty much a predecessor to the helicopter. For all his inventions and all the drawings of his inventions, he left detailed notes and measurements of how they would work. And they've led to modern day inventions such as a helicopter. And even now there's new instruments being made by artists, new vehicles that sometimes work or do not work, but extremely interesting things. And inspire. So the Jupiter poster is a series of posters from NASA titled Visions of the Future. It was created in order to inspire what NASA calls the architects of the future. They want to get people to be interested in the work they're doing and support it and create more on it and build on it. And this thing here, which is actually X-ray diffraction image of DNA, is another similar thing that inspired a lot of people. It doesn't really look like it. It looks like a circle and an X. But this is the work that led to Francis and Crick to discover the helical structure of DNA, which led to the revolution in genomics and caused people to be able to test for cancer, learn how diabetes work, treat the genetic diseases. And this is probably one of the most inspiring works of the recent century in science. So now I'm going to talk about what I'm really passionate about, art inside of a molecular biology lab. There is a lot you can do inside a molecular biology lab with art. And it's really hard to explain because it's pretty technical sometimes. So I'm going to just start with some of these images. So this image of blue, green, and red is actually the staining of neurons, which is involved in the circadian rhythms of the Drosophila. Drosophila is your common housefly. Circadian means your pretty much internal clock. And what it shows is the blue neurons, which is known as clock neurons, releasing a protein called, I believe it's neurotonin or leukotonin, which is illustrated in the red, to another cell that called leukotonin receivers that are illustrated by the green. But beyond the information you can get from this kind of picture and the function of neurons and circadian rhythms, it's also an extremely beautiful image to look at. It's actually the fly's face, and it can actually tell where the eyes are, the nose, the mouth, and how all these neurons are interacting with each other. The next image is actually a beautiful set of images created by studying embryo development using light sheet microscopy and computer tracking. So these images are actually digital. And the way they were developed was by taking microscopic images of embryos throughout development and getting a computer to drag each individual cell throughout its development and determine its fate. It then labeled each of the cells with different colors in order to determine overall cell fate. It's an extremely useful cell and tool in developmental biology, and is also an extremely beautiful illustration as far as I'm concerned to look at as well. As well as, have you ever thought about using C. elegans and performance art? For those of you who don't know what C. elegans is, it's actually a little nematode that lives pretty much everywhere. It is known to live in your soil, your pots, your plants, your food. I mean, you probably have eaten it many, many times, and it's kind of adorable in the way that nematodes are, I guess. But they're quite ingenious. There's one technique called optogenetics. I took it down because I had to show you the video, and doesn't really show in the presentation form itself. But there's a technique called optogenetics, which involves attaching a channel rhodopsin, which is a protein that responds to light, to a gene, and using that to turn the gene on and off during any point in time, just with a flick of a light. And in this study, they actually did it using a blue light, which turned on and off a gene called RCN, I believe. And of course, PowerPoint is absolutely wonderful. So you can see there's a blue strip on the C. elegans as it's moving around. And what it's doing is stimulating it to express that protein. And once that protein expresses, it induces the C. elegans to start laying eggs. Using these kind of methods, you can even cause them to dance in a sort of way, control where they move. You can cause them to go to where things that you usually don't want to go back from other things. You can cause them to breed like rabbits, if that's accurate. Or you can cause them to be very, very selective of their mates. And you're probably going to see it soon. It's going to start popping out eggs out of the sides. And that small little bumps are the eggs of the C. elegans. Something else you can do with C. elegans is create, this one was actually genetically modified with 3 different fluorescent genes. One is green fluorescent protein, another one is red fluorescent protein, and the next one is blue fluorescent protein. This was used to show where the DNA was, which is blue, RNA, which is red, and the rest of the cells are just expressing green fluorescent protein. And something like this is extremely beautiful to look at. It's actually not as this specific piece was actually done for an art contest. And it's extremely beautiful to look at, as well as it provides an understanding of what's happening inside the worm as it lives and moves on. So the next question is, what can art really do for science? From a scientist's perspective, what is the biggest contribution art can make? Well, first you can attack the obvious, illustrations. Pictures are pretty. If you've ever been in an undergrad molecular biology class, your diagrams are usually boxes with other boxes and lots of writing. Another one is something I like to call the fear of science. People are really, really, really scared of science. So how do you show people science is beautiful? And I think this is the biggest application where art can have. You can show science is beautiful, that it can be not scary, that's easy to understand, and it can open the concepts up for everyone. What can science do for art is the next question. Well, for one, you can try to understand what art actually does to the brain. For example, one researcher decided to test using Bio.TO against Parkinson's. So what this means is that he took Parkinson's patients, and Parkinson's is known as a loss of neuroactivity, where you can't really control your body anymore, and got them to take Bio.TO lessons. This was based on neuroplasticity in your brain. So this goes on, if your brain is flexible, your brain changes depending on your environment. It will change when you start doing Bio.TO. And they found with a significant amount that Bio.TO actually improves Parkinson's symptoms. You can also create new ways to communicate in art. You can send DNA to people that says messages. You can send a love letter on DNA. You can create a bacteria that does something very special. You can make it smell funny like bananas. You can make it express different colors during different stimuli. And you can also understand nature a lot more intimately. I'm not just talking about like talking to your frog and, kissing your frog and it turns into piss. I'm talking more like going inside the frog cells, and understanding why the frog does this based on this protein reaction with the DNA. But there is a lot of barriers. Lots of barriers. First is knowledge. How do you bridge the knowledge gap? It's the way to say it's not quite easy to just go into science. It's not the hobbyist thing you can pick up in a week. There's a lot to know. There's a lot to do. And how do you even do that? Skills. Where can you even get the skills? Who's willing to teach an artist how to do lab work? I mean, certainly professors aren't. They're too busy with their own lives. Grad students are trying to get their graduate degree. Who else can teach them? As well as access. I'm not sure how many of you ever tried to access a lab, but it's hard. You can't do it. Basically, you just can't do it. There's liability, it's protocols, it's safety. There's a lot of issues. And they'll just tell you, if you ever want to go in a lab and say, I just want to try to do something like this, they'll say, no, you can't. Just go away. So now, where do you begin? This field is moving. It just started recently, and it's gaining traction and it's moving fast by artist scientists and scientist artists. So we want to turn art into a science, as well as science into an art. So now I'm going to talk about what I do, what DIYBio does, and why we hope to make this, further this vision and make this more of a reality. So first of all, we're a group of people who decided that we're tired of hearing saying no to a lab. We want a lab to do our own work. There's various reasons why that happens. First of all, we don't own our IP if we work in university or industry. We lose all intellectual property of our work as soon as we step into their lab. We can't get access to some of the equipment. I mean, what university is going to let you use their mass spec for what you want to do? Or how about the PCR machines and the centrifuges? As well as people who just really want to learn. There's a lot of people who join us who have absolutely no science background at all and just want to learn biology, want to understand what's happening. Sometimes they have their own projects, sometimes they just want to work on something. So the next question is, why should we really exist? Why should there be a lab where people can do science? Basically for everything I've mentioned. We want to open it up. You want it to be less scary. You want people to understand what is actually happening in science. So what actually happens in the lab? I mean, I just kind of said a bunch of words you probably didn't really understand. So I'll kind of actually tell you more in detail what happens. For an entrepreneurs, it's a lot of working on their idea. You have one guy who constantly uses PCR to replicate his DNA. He wants to invent a new way to do DNA synthesis. Personally, I just like making E. coli do funny things like glow red or smell like bananas or anything that kind of interests me, basically. And there's others that they just kind of do experiments. They're just like, oh, like, tell me something to do, and I'll train them in the lab skills and the basic lab skills and what they need to learn. So who are the kind of people who join DIYBio? Is this really just a bunch of normal people or a bunch of like really crazy scientists? I would say we're kind of like more on the normal side. I mean, we're all super enthusiastic about biology. I will talk your ear off about anything. But on the grand scale of things, we're fairly ordinary people. You have anywhere from PhDs to absolutely no education. I have people with only high school degrees, and I was teaching them how to manipulate E. coli. We have people with PhDs working on their own projects for our company. It really varies, and there's never been any friction between the different education levels. We're just kind of accepting, and the lab is kind of nice because it's very creative. There's balloon molecules and other sort of exciting things, skeletons, dinosaurs. Well, it's tiny dinosaurs, not big ones. And we also like to mention we're actually sponsored by the Hack Lab. We're located inside of them. Hack Lab is pretty much the paradise for nerds. And by nerds, I mean suing nerds, music nerds, bio nerds, any kind of nerd, really. It's a very accepting place. No one judges you based on your sex, your sexuality, your orientation, your background, your culture, nothing like that. And they're all fairly, from the extremely ordinary people to the extremely, extremely nerdy people, we're all just fairly talkative and inclusive. And that's the end. So if you want to contact us, there's an email, or if you want to visit us, this is the address. We're open every Tuesday evening for open house from 6 till late. And that's it.
Turn the light on, or? Okay. Good. Hey, so this is actually the fun part when we start asking questions and we start the conversation. So, and we have two microphones, so we can stand and respond, and so we have this one and that one. And the audience will need to scream or will need to repeat the question. So otherwise you won't come up in the screen. Okay, so does anybody have a question? Yes?
Jasmine?
Yeah.
General science. Does that mean that unscientists ever people excluded or not look like nice among them or something?
No, it's more like we have safety standards we creep up to. As long as they are within the safety standards, we're fine with them. We just never really had anyone's absolutely crazy come in like, I want to do this sort of thing. We did have a couple of people ask a couple of weird questions that were not quite the best thing. But for the most part, it's not like they're unincluded. It's just most people who come to this place are not usually the mad kind of scientists.
Where the cool stuff is.
It's also the most dangerous sometimes.
Well, one of the biggest inventors of all time were killed in accident, but accident. So if you like right by the older safety rules, you'll never do my science, you never like make progress, you never go good. But it's science. Anyway, because I consider that much science bullshit, I want to like do stuff, invent stuff.
Yeah. It really depends on your definition of what you're going to do. It's just be safe about it. That's the biggest thing. The safety thing is a serious concern. And the reason we want to open the space up so people don't do this kind of stuff in the labs, because some of the things, the reagents you can't deal with without the proper facilities. such as high concentration in HCl or like other stronger acid bases, surfactants, stuff like that. We want them to be able to do it in a place where they can do it and not pass out from the fumes. Right.
So there's one
How much does it cost to access the DIYBio lab?
So our membership is only $50 a month. It's just a flat fee. For the work itself, it's we have a lot of communal reagents that you can use because it's a lot cheaper to get like 1 kilogram of pure salt than it is to get little packets. And for the rest, you just kind of pay for your own experiments. So if you're doing analysis on certain DNA sequences, you just order your own DNA and stuff like that. We have accounts with various providers, and they're more than willing to give us what we need.
Are these visiting the lab?
Yeah, sometimes I come visit a lab. We don't have artists in-house yet, and I'm hoping, really, really hoping to change that at some point in time.
That was actually one way to invite artists to DIYBio. Because I thought inviting artists, and I'm actually going really often to Art Lab every Tuesday. There's an open house and they are cooking for everybody. They went 10, so I'm one of the cook. And so I've been talking with people, and I really want artists to come in and do some projects, like photographic people. Like I've been actually thinking about photo shooting and stuff like that, and it will be very interesting to see what comes out of it. Like a little mix and match. But Elaine had a question.
I'm interested in the notion of contamination and how it works for both the DIYBio lab and how you adjust for that. And then Nicole, you're actually working with it when you decided not to work in a sterile situation. Can you both talk about that?
Yeah, so I kind of came to realize the impossibility of it in my situation. So in a studio environment where I'm also making other work. I'm not just exclusively working with bacteria. I'm not setting up a completely sterile open space. I'm also grading in my studio and doing other activities. So just trying to keep contaminated didn't seem plausible, but also I obviously could seek out a space like DIYBio lab or try to set up a corner that's sterile, but I think to kind of appreciate the lack of control to a certain extent that I was working with normal flora. I mean, you've been to my studio, and most recently I've been working with slime mold. So with that, there is a little bit more sterility necessary, but yeah, so there's some gradients where I can get to a certain extent of keeping plates sterile, but definitely not, I'm not particularly interested in being able to isolate particular things necessarily.
Would you move and try a laboratory with a sterile place in that condition?
I would try. I mean, I think I would explore it, but I'm not sure. I'm not obviously compelled enough to be actively switching. Yeah, so I think I would never say no to an opportunity to try it out, but at the present time where the work is going, I think it is working to have that kind of studio environment.
Lab sterility is always a problem in labs, especially in our lab where we don't have a big gigantic autoclave. So we kind of sort of like figure out how to do it ourselves. So one thing we do a lot, we have a lot of bleach, tons of bleach. Lots of alcohol. We also have what I call is my mini autoclave, which is technically really just a pressure cooker, but it works and does the job phenomenally. The biggest thing we practice is something called aseptic techniques, which is where we culture bacteria in such a way that minimizes contamination. So using the pressure cooker, we sterilize all the components we need, we plate it, and then when we plate our bacteria, we make sure the plates stay closed most of the time. We also have a glove box available made by one of the members where you can actually put in an argon inner atmosphere in it if you want to work with a completely sterile area. But for the most part, we find with aseptic techniques and biological safety level 1 work, which is your basic kitchen level, it has been proved to be effective for the most part.
What steps are required to make things well? The colors are obviously pretty exciting to keep that culture weird things, but Actually, how many steps do you have to go through? How long does that take you in time?
Well, surprising enough, it's actually really easy. It'll take you about a week to do if you do everything on time and your experiments obviously don't fail. And what you do is what you can do is order the gene which is usually just gluing fluorescent protein. You order the gene, and for C. elegans in particular, what you do is you actually change its food. So C. elegans eat E. coli. So what you're going to do is you're going to modify the E. coli, which is basically just by shocking it really hard in order to make sure they take the DNA up inside of them, and then they start glowing. And then once the C. elegans eats the E. coli, they're going to absorb the genes from the E. coli. Don't ask how this works. No one knows, but it just happens, and it's pretty much like magic, and we're just kind of grown to accept it. And once the worm eats the E. coli, it will actually start expressing the protein itself. Yeah. Yeah. We used to have about 10 different colored E. coli cultures for fun and to paint with.
Would you be interested in doing a course for people teaching us to do these kind of things?
We're actually talking. There's actually going to be a course coming up soon led by a guy called Justin Pereira, and he's going to do some cell transformations, teach people how to make different colored E. coli at the lab. So it's going to come up soon. We have a meetup page, and you can just keep an eye on that or our website to see when it's happening.
First of all, I want to thank you both, because I was kind of wondering what's the science and art connection before coming here, but now I have a pretty good understanding of very symbiotic. It's obvious. And also think, now that I understand what is common to both is life itself, and both are concentrated on life very heavily. So I had a question. It's not a scientific one because I'm not a scientist, but if anybody remembers the movie The Biologist, and the architect says, art can be dangerous. So when I was looking at Nicole, some of those things, I was thinking, how close can we get to this? Is it dangerous? I think it's a question for both of you, but can it be dangerous?
So I'm also operating at a
Lab 1 level, so I'm not culturing anything infectious. So basically, I still take the utmost precautions, so no one is able to touch the work, right? So there's always a vitrine, or as you saw with the mannequins, it has a plexiglass case. I like the idea of art being dangerous in a more conceptual way. So I think my art can be dangerous, but maybe more ideologically than physically.
For DIYBio, it's the same issue at this first safety level. There's basically what's classified as little threat to an individual and little threat to the environment. And everything in culture is anything you can find in the common house. If you looked at, let's say, your shower drapes, you'll find lots and lots of cultures of bacteria, for the most part, which are fairly harmless and non-pathogenic. It's only when you start culturing human pathogens, which is a higher biological safety level and is monitored by Health Canada.
What is a human pathogen?
Herpes. Yeah.
yeah.
DIYBio offered the definition for art, the definition for science. How did that look crazy with footnotes? I'm not sure which. To me, your definitions, number one, where did you get them? Interchangeable.
I know, they're terrible. They're pretty terrible. I got them from Merriam-Webster Dictionary, and I just wanted to see, I kind of was wondering what is the actual definitions of these two subjects because I never looked it up. And I want to see how do people actually clinically view art and science from outside the field. So that's why I chose those definitions. And when I read them both, I really did not want to use them. Yeah.
Actually, discussing is that so I'm listening to hear the more ideas of the concept, especially about between something. It was clear that we got to see the woman that you are office science to know that art is not about beauty. That it's not a beautiful thing. For example, when I make artwork, and when I work with the critics, the best criticism about it is your art was bad, and you always have good criticism. And so these days, people think that suddenly it's a substitute, like a kind of a political requirement. But coming forward from your artwork, I participate. Then suddenly you can go all the way down. So I just want to know more.
Sure, I think there is this strange phobia in the art world of the word beauty and of things being beautiful, and that somehow makes them not conceptually rigorous, which I think is absurd. And I mean, I personally am actually starting to use beauty strategically. As was sort of mentioned, science can be this monolithic, intimidating subject matter. And art, similarly, can be this hugely intimidating subject matter for an individual to come up to an artwork and they're almost they feel like they have to know what it's about, right? So there's an intimidation factor on both of these fields, which I'm trying to work with. And so I'm using beauty as an entry point. You could approach the work and find it beautiful, and then slowly it can reveal itself conceptually as being involving bacteria or mud or having scientific aspects to it, but it doesn't present itself immediately necessarily as this kind of really rigorous object. And then also just thinking about like a much larger discourse around the sublime, of beauty not being sublime, of like Kant talking about the beautiful versus the sublime, and Burke as well as the separation. So it's kind of like an interesting territory to be traversing, that the work can also be beautiful and sublime. So I think I'm still working through that conceptually, like how that can still work. But generally, I would say that the sublime can be beautiful, but the beautiful, if something's labeled just as purely beautiful, it cannot be sublime. So it's kind of this strange space to be in, but I think there's some area in there to work through, yeah. That's tricky, yeah. So I think that, let me think. The beautiful, right, so if we're thinking about it in terms of Burke, the beautiful would be something that we appreciate with pleasure, right? So pleasure would be the first response in terms of the discourse surrounding the sublime, so speaking to Burke specifically, that you as an individual would gain visual pleasure from it, right? And then the sublime, you have pleasure, but then it's undercut with terror. So it can at first appear as beautiful or have beautiful qualities, but it has other qualities as well. So that's kind of the distinction I'm working with under the discourse of the sublime, but more subjectively, I think it's very elusive what is beautiful. Yeah.
Question, Nicole. You mentioned slime mold.
Mm-hmm.
I remember reading papers going back to the 90s about slime mold. One of my favorite scientists thinks it's intelligent. One of my favorite philosophers think it's intelligent. So I would like to know your opinion about it. Is it intelligent or not?
That's a very interesting question. So the research I'm currently doing is kind of investigating how we value slime mold. So for anyone unfamiliar, slime mold is an organism that's capable of solving mazes. It's been applied, there's a good TED Talk on it by Heather Barnett, who's also an artist, but it's been applied to map out the Tokyo subway system. So it's being applied analogous to a team of human engineers is a quote that's often used. So it's often being compared to human intelligence, and I personally find that completely absurd, that we would take an organism and only value in it what we personally as humans would find useful. So this idea of intelligence, I think it does have intelligence, but I think its intelligence is its own, and that we're kind of using it. We're using biomimicry to kind of take what we think is useful of it and only valuing that. We appreciate it for doing human things better than humans, whereas I think that's kind of just a really strange way of valuing an organism and probably reflects upon our relationship to the environment in a broader way as well.
So this question is for both. Through your journey as a professional and just through tinkering, what have you discovered or realized with biohacking that kind of relate or apply to your life? It's like aha moments that what seems as kind of abstract, but you can actually see and kind of push it.
Well, from my experience of working in a lab in DIYBio, a lot of the times I kind of encountered disbelief or sort of like people are like, you can't do that or don't do that or it's not even possible. And then actually getting it done, you think, oh my god, you got it done, it's amazing. And then you realize, no it isn't, because now you have a bunch of paperwork to do. But it kind of made me realize in a way that the things, I guess for me it's kind of cheesy, but the things you kind of want, the things you want to accomplish, right? It's not going to just happen. And if people tell you not to do it, it doesn't mean it can't happen. And that regardless of what you do, you should try and keep trying. And this is the thing with science. You keep trying, you keep repeating experiments until you succeed. And if you don't succeed, you try something else.
I would definitely say it's
more, again, going back to this idea of the microbial sublime. And for me, my moment of really feeling that presence on and in my body, of realizing that I'm not made up of exclusively what I thought I was, and how that made me start thinking about my relationship with the environment and with just even like our immediate environment, not necessarily environment as we are typically or stereotypically understanding it, that I'm permeable and you're permeable and that our microbiomes are constantly mixing and we're not separate. I think that's probably...
Following up on that, how do you feel about leukocytes wandering around in your body, like independent of no agents and blood corpuscles going through your body, the number of blood cells moving or exceeding the number of cells or other productivity? That a similar creepy area, or in fact, it seems to be different than the microbiome.
Mm.
But it's equal
Mm-hmm. Yeah, definitely. Definitely. I think this idea of, anything that is within us that is kind of, and almost all of our body actually operates in this way, so it's a little bit tricky to make distinctions, like, oh, the bacteria is sublime and the blood cells aren't sublime. But, I would definitely say that any sort of level of contemplating how your body operates can become a sublime experience. That even thinking about your breathing and how when you start to pay too much attention to it, it actually becomes work, and you have to think of like taking your next breath in contrast to your body automatically doing it. And that experience of control versus lack of control and automation, I think all of that is quite an interesting area. Mm-hmm.
Our bodies and having other. We don't think of ourselves as other species.
Mm-hmm.
The mitochondria has captured bacteria. Yeah. And every cell in our body is well. It's just the exchange, ourselves dying and replaced. That whole cycle, we're not the same people that we were last week.
And then after that, there's another person. So,
that leads me to the interest in the DIYBio artists. I learned that it was the burning notion of art, and if you're actually working with living organisms, and that you have to, is there an ethics? Here, there's always a bioethics that's going on, and the way we approach working with other organisms, because these other species are part of us, but often they're not recognized as other species, and not necessarily those questions that come around working with them. Does that play into what happens in the laboratory and
Yeah, I mean, we have always many questions about certain rules in DIYBio. In labs, very often you're not allowed to culture any kind of human cells at BSL1, which is kind of odd because technically you're culturing cells every single moment you're breathing. And when it comes to dealing with organisms, it's a lot of scientists, when it comes to dealing with organisms, they kind of get desensitized, especially when working with animals. And one thing I kind of like to do at the DIYBio is that even though we'll probably eventually allow larger organisms in, you mostly deal with microbial as well as small nematodes. And it's the biggest question you're going to have asked if we deal with this, and when we deal with these organs, how do we deal with them? When we're done with them, what do we do with them? Do we share them? Do we destroy them? Or do we keep them preserved indefinitely? And it's always something we're always working to answer, and we don't really have an answer for it quite yet.
Yeah, I think there's constantly a consideration of it. Like even the fact that working with bacteria, if you're working with it, you're eventually going to kill it, probably, for safety reasons. But also even in my work, as the agar dries out, the bacteria are dying, and I've sort of given them life and now death as well. So there certainly is a consideration of that, of the valuing of a microbial life, which I think I am contemplating, and perhaps there's a work or there's some sort of text there to discuss it. Also with the slime mold that I'm working with, I'm trying to really, this is I think another layer, thinking about them as collaborators and trying to sort of value that and not just use the organism. So I'm still, again, negotiating that and living with the slime mold in my studio and trying to come to an understanding of it and its desires, like what questions it's interested in being asked in a way. So I think ethically there's considerations there of how do you collaborate rather than just impose upon these organisms.
Yeah, there was somebody in the back. I have a quick question and a difficult question. The quick question is, you said something about 10 times the mass. You're saying that they're in our body?
It's not the mass. It's 10 times more microbial cells than human cells. Yeah, in our bodies. Oh, 2 times. Okay, wonderful. Yeah.
All the time. Just nobody has considered before.
And then a more complex question is, you talked about beauty as being an access point to engage in this. I'm also wondering how, you mentioned it briefly, but how disgust might also be an access, like little kids just love popular YouTube series about gross science, right? So have you used that or like?
I've found disgust is I find it less accessible. I think that it's present. I think even if I tried exceptionally hard to get rid of it completely, it would still be present. Working with biological matter and bacteria and microorganisms specifically, I think comes with a certain level of people getting grossed out. With the mannequin series or work, I found that actually to be less successful than the sheets because of the figure and how horrific that can be. People got Chernobyl victim from it. It was a child-sized figure. So there was a lot of disgust or abject surrounding that work, and I found that made it much less successful. I mean, I think to a certain extent it did, but it's not a slow interest. And I'm interested in slowness to a certain extent and spending time and allowing for speculation, which I think disgust can become really spectacle-based. So you arrive at it, you're disgusted by it, and then you retreat from that disgust. But definitely I think it's certainly a strong emotion and definitely a strong reaction in the viewer and a strategy that absolutely can be used and is used by a lot of artists, just not one that I'm personally interested in continuing to employ.
Would you
see sort of as the potential for, this is a question for both of you, just for the expansion of this, given that there are certain, not so hard, but limits that are there about what we can do, whether it's what do you see as a strategy for making diverse artwork, given that most people have really small rules of putting into disposal? I see a lot of stuff that comes out of looking careless that people are doing when I'm looking at bio and also making electronics kind of the same sort of thing with the whole stores that everybody buys electronics. So
I think that's actually quite difficult to answer. So a lot of the images I showed you are more incidental from the research than the intention of the research. And getting something that looks sufficiently different and sufficiently against bio art from what is already present in the day will take some almost extremely ingenious way of combining skills. If you can use a tool such as CRISPR, which we can do in the lab, genetic modifications, or modify a sufficient amount of genes and C. elegans to make them do unique things, you can get something that is a lot different from what is in the present day, but also be appreciable in more of the minute details rather than the whole picture. So it'll be learning to understand and see those details as well as combine things in such a unique way that you can actually create a piece that is different from what is already present.
I would actually say this is a challenge of all mediums. So I would actually be much more intimidated to become a painter right now with the weight of the history of painting and with obviously the diversity of work that's already been done. You could say, oh, I can't do anything new with paint, right? So I think there's a similar limitation with all materials, and you are going to employ the material that you think is most appropriate for the work. So I'm not going to limit myself to only working with biological matter if I feel perhaps that the concept is better evoked through plaster or a combination of art materials and more bio-art materials.
I heard the term bioethics a few minutes ago, and I'm not trying to start a debate here, but ethics in biology, or actually I'm just going to say science, generally, very difficult things to put together these things.
This, I think, is for me. However, Nicole, you said earlier, or Jasmine, I can't remember which one, but you said that art is a language that transcends a whole bunch of things that we are normally daily likely use. Now, could art be used to address this concern about the ethics, the understanding of the ethics in the science that I personally can't wrap my head around? Going back to an example about the sovereign system, the organism, and the ethics related to that, you know what I was thinking? Well, there are some people in this room probably look at a chicken and they don't think of food. Yet we breed them for that purpose. And yet we breed dogs, for example, or we just for pleasure or companionship. And we do make those selection and decisions, even in higher organisms. But when it comes to certain, I guess, arguments, we can easily slip into kind of a unclear whether or not I think science would be a good language to explore that. What do you think?
So, sort of to address what you first said about is art capable of addressing these ethical dimensions of science, I would say absolutely. Particularly because we have a position of being slightly exterior. So as an artisan brought up through, who personally has been brought up through an arts education, I'm not as engrossed in a scientific education, therefore potentially have a perspective that wouldn't necessarily basically I'm able to look at maybe the practices of science in a very different way than someone who's engrossed in sciences. And similarly, of a scientist looking at my practice, they would be able to point out the language that I'm using that's esoteric, that I just take for granted as part of, talking with other artists. So I definitely say there is the possibility for reflection. In terms of you're speaking about sort of selectively breeding animals in relation to ethics or using animals for food. I think that when I was talking about slime mold, I would like to apply that to a much broader understanding. So yes, I'm speaking specifically about one organism, but through cultivating a collaborative relationship with that organism, I hope to speak to the way we treat all organisms to a certain extent. So although I'm isolating and speaking about one thing, I'm kind of thinking about our relationship with the environment and organisms and our utilitarian value sort of way of thinking about them as a whole.
I think what Nicole said describes exactly what I was saying. So.
I like Nicole's stance on this thing, and I share it completely, but the cold reality of it is that all this bioethics stuff is driven by market. In Korea, there are already factories running cloning photonic paper. You just pay, you get what you want. It works. People are happy. The police department to sue all their family dog, the best worker dog, we clone so they get more money out of it. So it's all market, it's all money. There is no bioethics per se, like something intangible. It's just money. Pay, you get what you want. That's just what to say. But all we who believe in art and intangibles and philosophy and all that, that's all good. But unfortunately, that's not how it works.
I'd still say that in individual ethics is important.
It is.
Yeah.
So, although the market may obviously drive what is and is not allowed by governments to a certain extent, individually feeling strongly about a certain bioethic, and if all of us as individuals did have a stance on this, we certainly could impact the market value of that as well. Well, with genetically modified organisms in the EU, it was definitely impacted by public discourse. So I'd say there certainly is a limitation, but a potential for...
But we assume that we, the people, know what is going on, but that's not how it is. I happen to read Russian research in all this area, and sometimes older people, like professionals, come, and they share details that just reading what they say, you understand that these are real scientists, that are not fakes, that are not teenagers, people who have no clue. They're completely real hardcore scientists. They used to work for research institute in Siberia, like completely secret institute, you don't know ever that they even exist. The stuff they were doing there, I just horrible. Most of the people I read and say, oh, let's stay away from me, you're evil. So you don't know what they're doing, and it's not just nationalists. I suppose that every country is doing this, so we don't know what they're doing, but they are doing it. So what we believe is always good. It's good. Doesn't matter there. Anyway, I just want to say this.
So actually, I have a question. I'm actually very curious about. Because you mentioned a little bit the question of intellectual property, and when I'm thinking about art, I'm thinking about authorship, and you were mentioning the collaborative aspect of it. Collaborative, like we think collaborative because we cannot actually talk to the microbes. So I'm trying to put together the two things because I remember the few talks that I heard at the beginning of the making of the DIYBio.
movement here in North America. I don't know about other places. My understanding is that it's different, but like there's a lot of obsession for intellectual property. Like it's your work and you don't want to, I mean, you want to share it, but you don't want to share it at the same time. So it's a little bit of like a weird situation that is going on. And it's the same with, and then I was thinking it's the same with art, because yes, you are trying to collaborate with your organism, but then you still try to make the organism do something. So like I'm trying, like, I would like you to say something about it, because I find it intriguing.
Yeah, from the DIYBio perspective, collaboration is like, IP is a complicated issue. So the whole thing started because we want to open up science. There's a big push by academic, researchers, industry, basically anyone who wants to read research to make all research public. And because of that, you get sites popping up that allow you to access all these scientific papers for free, which, governments always want to shut down because infringing intellectual property and stuff like that. And the biggest thing for DIYBio is that for people who want to own their own IP, there's no available way to do that. So what we do is kind of give the freedom for people to decide what they want to do with their property or not, if they want to keep it or if they want to share it. And if for most of us, it's just at least giving them the option to decide that. Because if they went to industry or went to university, they don't get that option. So it's more just giving the freedom of that.
Yeah, I think in terms of collaboration, I'm
actually really interested in relinquishing a certain amount of control to the organism. So certainly I'm setting up conditions. That's definitely absolutely accurate. So I'm setting up a framework for the system to operate within. But I'm not particularly interested in pinpointing exactly where necessarily certain cultures are growing or really controlling how the slime mold is traversing its environment beyond its own systematic way of operating. So I think I'm definitely interested in collaborating, and the fact that through this type of collaboration I'm able to create imagery or to work in a manner that I never could do just by drawing out, say, the path of the slime mold. I'd never be able to think in a slime mold way enough to be able to do that. So I think through not having complete control, the work goes beyond what I could ever do.
Another question that came out of that. Oh, you have one. Sorry, I don't want to take over. So yeah.
So where do you find these open source research papers?
There's this website. I'm trying to remember the name right now.
It's this Sci-Hub?
Yeah, Sci-Hub.io, is it? Yeah, Sci-Hub.io. Most of it's in Russian. Just ignore the Russian and type in.
Because it's been no longer than me. So actually, from a DIY scientist, where there's also DIY biology. We're actually it's not plant biology. It's not legit reason lab that is working with art. And I had this long discussion with him, and he said, no, it's not good that everybody's talking about this science, because then all these people who do not have real interest in science are going to go and download papers, and so the site is going to be shut down. But this is not the case. So it's kind of discussed.
Yeah.
You're going to find it. It's been all over.
Yeah. And people sometimes have the theory if they share the knowledge too much, it just loses value.
The question of sharing is actually like contentious right now. That's why I've been asking about the intellectual property. And also, like recently, among the citizen scientists, there was this discussion about the fact that a lot of people are sharing their own knowledge, but then here comes the entrepreneur, takes the idea, and then turns it into a kit that then is Indiegogo or crowdsourced, and then they make money out of it. And some people don't like that. So at least these kind of things.
It's a fear in the arts as well.
Yeah.
As an emerging artist and a more potentially successful artist who was to take up the idea and proliferate it. I mean, I think there's similar fears, but I can't implement with these insecurities.
There's all.
This would have been who Wilson now nobody knows who invented it. But then it's stolen, then that's it. And nobody cares anymore.
But it's always been like this, actually.
Yeah, so what's the solution?
And, we pretend to regulate, and we keep talking about regulation. So questions for me are like, so what do we need to regulate? Is regulation important and or not? So it's
information is not really so much worse because progress is slowed down, and it will stay forever in the dark days.
In my personal opinion, if I had my way, all knowledge would be free to access, and like that would be ideal for me. But that's not the case, and I guess one of what you're saying about people giving an idea to someone else taking and cashing out on it, it happens. It sucks. And yeah, and I think that's one thing you have to come to to accept as the risks for sharing information freely.
More comments? So thank you very much. That was a very interesting conversation, and I hope we go on. And please do visit DIYBio. When is the next event?
Actually, I'll be there probably Tuesday from 6 to midnight. Well, I probably leave before midnight because I live far away, but I'll be there from 6 on. Yeah.
Nicole, are you gonna have any exhibition upcoming or?
Nothing firm. But if you check it, just Google my name, check it on my website.
I put your website
amazing.
On the website, on our website. So if you go to artsalon.com, you can find the links to Nicole's website, to DIYBio lab, etc. So thank you very much.